Hello, how are you? Today, inshallah, we will complete with the recombinant DNA, part 3. For the last lecture, we mentioned how the recombinant DNA technology will form, how we can use the recombinant DNA technology in formatting or forming a new gene, and how we can insert specific gene in another gene. And we mentioned how the plasmid DNA is a suitable host for the recombinant gene or for a small DNA fragment. And we mentioned that if you want to make a recombinant DNA, we need to use similar restriction enzyme. And we mentioned that EcoR1 is specified for a specific sequence, and once we cut with this enzyme, the resulted sequence will be similar. And in this case, once the resulted fragments have a similar sticky ends, then there is another enzyme called DNA ligase will bond these two sticky ends together to form a new DNA. Once the new DNA is formed, this is called recombinant DNA. For today's lecture, inshallah, we have a specific object which is define transgenic and describe the usefulness of some transgenic organisms to humans. So today, inshallah, we will use the transgenic organisms, plants or animals. So how can genes from one organism be inserted into another organism? Can you imagine how we can insert small gene in another organism? Or how we can take small gene from specified organism and insert it in another organism? So transgenic organism can be produced by the insertion of recombinant DNA into the genome of host organism. So we have to form the recombinant DNA first and then insert this recombinant DNA into host organism, okay? This is the new technology of the transgenic organism. The universal nature of the genetic code makes it possible to construct organisms that are transgenic, containing genes from other species. So we have a transgenic organisms which contain specific genes from other species. Transgenic organism can be produced by the insertion of recombinant DNA again into the genome of a host organism, like the bacterial plasmid that we mentioned in the last lecture. So the bacterial plasmid contains specific gene, and it contains also specific genetic marker, and that will be inserted in another organism like plant or animal. Transgenic technology was perfected using mice in 1980s. So in the beginning, they used it on the mice, and genetic engineering can now produce transgenic plants, animals, and even microorganisms. Can you imagine? Look at the picture here. Here we have transgenic strawberry, and here we have transgenic animals, and here another transgenic mice. So this is how we can form or how we can combine between the recombinant DNA technology and the transgenic organism. Many plant cells can be transformed using agrobacterium. Agrobacterium, specific bacteria for plants. In nature, this bacterium inserts small DNA plasmid that produce tumors in a plant cell. So the main function of this bacteria is producing tumors. So once the bacteria produce a tumor in the plant size will increase, okay? So scientists can deactivate the plasmid tumor-producing gene and replace it with a piece of recombinant DNA. Sometimes this piece of the recombinant DNA will be to increase the size of the plant, or to change the plant type, or even to add a specific characteristic to that plant. The recombinant plasmid can then be used to infect and transform plant cells, and the transformed cells can be cultured to produce adult plants. So in the beginning, we will take the agrobacterium, and we can remove or deactivate the plasmid tumor-producing gene, and then add another gene, a recombinant DNA piece, and once we added the recombinant DNA piece, we will grow it and infect the plant with it, and once we infect the plant with it, or we insert the we insert the recombinant DNA after the infection, the plants can be cultured. And by this method, we can form the transgenic plants. Look at the picture again. This is the recombinant fragment or plasmid, and this is the gene to be transferred. And once they change it here. This is the gene that wants to be transferred. And once we go back and insert the recombinant DNA or the recombinant plasmid in the bacteria, then the bacteria will infect the plant cell colonies. And once the plant cells colonies is infected with the plasmid or the bacteria, the bacteria will insert its genes inside the plant, and once the plant is infected, we will grow the plant to form or to produce an adult plant. There are other ways to produce transgenic plants as well, not just by using agrobacterium. When their cells walls are removed, plant cell in culture will sometimes take up DNA on their own. If we remove the cell wall of the plant cell, okay? The cell composed of cell wall, cell membrane. If we remove the cell wall, the plant itself can take up DNA by its own. And DNA can also be injected directly into some cells. And by this method, transformation is successful. If the transformation is successful, the recombinant DNA is integrated into one of the plant cell's chromosomes, and then the transgenic plant will occur. And we will get transgenic plant that we can grow it, and we can obtain a specific characteristic for the plants. Another method that they will use, which is transgenic animals. Scientists can transform animal cells using some of the same techniques used for the plant. As you can see here, we have a specific gene from the humans that could be inserted into the animals, and then the newly formed organisms will contain these genes. The egg cells of many animals are large enough to we can insert in it the DNA, or the DNA can be injected directly into the nucleus. And once the DNA is in the nucleus, some enzymes that is found in the nucleus, like the ligases and restriction enzyme like EcoR1, will work to form the recombinant DNA. And once the recombinant DNA is found ready in the nucleus, it will be inserted into the DNA or into the chromosomes, and then we can find the recombinant DNA inside the host DNA or the chromosomes. Recently, it has become possible to eliminate particular genes by constructing DNA molecules with two ends that will sometimes recombine with a specific sequence in the host chromosome. And once they recombine, the host gene normally found between those two sequences may be lost or specifically replaced with the new genes. And once we replace it with the new gene, we can form another characteristic. This kind of gene replacement has made it possible to pinpoint the specific function of genes in many organisms, including the mice. As you can see here, the cloning that we took it in the part 1 of recombinant DNA is a method that we can by it form transgenic animal. We can take a donor nucleus, and at the same time we can take a donor egg cells. We can remove the nucleus from the egg cell. Once we remove the nucleus from the egg cell, we can insert the nucleus from the donor nucleus into the egg cell, and then we will form egg cells with a specific or with the donor nucleus. And after the replication, the embryo will form, and then we will get, once the embryo is formed, we will inject it or we will implant it in a mother, and after the delivery, the embryo, which will be similar to the donor cell, not the donor egg, because we removed the donor egg nucleus and we add the donor nucleus to the egg cell. And this organisms or the transgenic organisms or the cloned organism will be similar to the donor nucleus. And the first cloned sheep is called Dolly. Don't forget this information. Dolly, the cloned or the first cloned sheep. By this point, we finished already the recombinant DNA technology. I want you to memorize all of the information is mentioned in the 3 videos. Please make sure that you will remember what we mean by recombinant DNA, what's the enzymes that we will use, and how the recombinant DNA will form, what's the types of plasmids, the genetic markers, and the transgenic organisms. For this point, thank you and have a nice day.